Hello and welcome to the Chancellor's Lecture Series. I'm Daniel Diermeier, Chancellor of Vanderbilt University. Today's panel discussion is hosted in partnership with Vanderbilt University Medical Center and the Vanderbilt Alumni Association. The topic of today's discussion is a timely example of the mission of the Chancellor's Lecture Series to connect the university and audiences across the nation and world with leaders who are solving the most pressing problems of our time. At this time last year, we stood at the brink of a devastating global pandemic, which has since then taken millions of lives and led to unprecedented upheaval. But now, with the worldwide distribution of effective vaccines and treatments that have been developed in record time, hope is in sight for an end to this crisis. The scientific breakthroughs of the past year would not have been possible without the pioneering work stretching back decades by the experts on today's panel, along with their colleagues at Vanderbilt and at other leading research universities. Our discussion today will be moderated by Provost and Vice Chancellor for Academic Affairs, Susan R. Wente. Susan's leadership as a leading biomedical scholar and as Provost and Interim Chancellor has made a profound and lasting positive impact on Vanderbilt. In July of this year, Susan will become the 14th President of Wake Forest University, a well-deserved honor. The three distinguished speakers on our panel also have unique ties to Vanderbilt and have each made groundbreaking contributions to COVID-19 research. Dr. Bonnie Graham is Deputy Director of the Vaccine Research Center and Chief of the Viral Pathogenesis Laboratory at the National Institute of Allergy and Infectious Diseases at NIH. His career in immunology began in 1979 with 2 chief residences and an infectious diseases fellowship here at Vanderbilt. He earned his doctorate in microbiology here in 1991. Dr. Graham's work in deciphering the immunological responses to HIV has dramatically advanced our understanding of antibody production. He recently played a leading role in the development of the Moderna vaccine for COVID-19. Dr. Mark Dennison is the Edward Claiborne Stallman Professor of Pediatric Physiology and Cell Metabolism and Director of the Division of Pediatric Infectious Diseases at Vanderbilt University Medical Center. He began studying coronaviruses long before our current pandemic, when support for these research areas was much harder to find. So it is only fitting that Dr. Dennison oversaw the first clinical trials of the Moderna COVID-19 vaccine, aided by the $1 million donation Dolly Parton made to Vanderbilt University Medical Center. Dr. Kathleen M. Nuzil is the Myron M. Levine Professor in Vaccinology and Director of the Center for Vaccine Development and Global Health at the University of Maryland School of Medicine. She's co-chair of the Coronavirus Prevention Network that has conducted the phase 3 trials of the COVID vaccines. She's also a distinguished Vanderbilt alumna who completed a residency in internal medicine and a fellowship in infectious diseases at the Vanderbilt School of Medicine before earning a Master of Public Health. I'm honored to have our panelists here with us today and will now turn it over to Provost Wente.
Thank you, Chancellor Diermeier, and thank you and welcome Dr. Graham, Dr. Dennison, and Dr. Nuzil. I greatly appreciate your taking the time to be with us today and for your tremendous contributions in the worldwide effort to end COVID-19. Thank you. And moreover, for the decades of prior work necessary to achieve these breakthroughs. I do think and hope that this historic pandemic has given society a new appreciation for science and medicine. Over the long past year, we've rightfully applauded healthcare workers like those at our Vanderbilt University Medical Center and physicians like yourself, who've worked selflessly around the clock and on the front lines to save lives. And as a biomedical researcher myself, I know how critical your efforts have also been in our understanding pathogenic viruses and developing effective ways to treat and prevent their spread. I also know how your work on this began long ago. I'm especially proud that Vanderbilt has a long tradition of success in this field, going back decades, centuries even. For example, back in the 1930s, Dr. Ernest William Goodpasture pioneered the use of cultivating vaccines with widely available chicken eggs. I know we're all eager to learn from our distinguished panelists today, so let's get right to the discussion. It's great to see all of you on this same screen. It's really impressive, and I'm so much looking forward to our discussion. And why don't we start off, I think, with a discussion of how the study of rather obscure viruses in the remotest corners of the world is immensely important to meeting the challenges of diseases that might unexpectedly emerge around the globe. Would you like to take on that long or question / beginning discussion, Dr. Nuziel?
Sure, I can get started, and certainly Barney and Mark will have a lot to say. You know, as you've said, little known viruses that emerge anywhere in the world are not irrelevant. And this is not the first time we've seen this with coronaviruses. It's actually the third time we've seen this in the last 20 years. And just last month, we had an avian influenza infection, H5N8, that infected poultry workers in Russia. So we understand that these viruses have the ability to transmit between humans and between animals. And if they also acquire the ability to spread between humans, then we have the situation that we have now with COVID-19.
That lays it out pretty clearly. I don't know if anyone would like to add further to that.
I just like to add one thing, and that is that even though the number of viruses that infect humans goes up in a linear way, you can show that the number of virus families that infect humans has plateaued. And that's one of the concepts that I hope we can talk about, this idea that this is a tractable and definable, big but doable problem to really focus in on those 26 virus families that can infect humans and do what we call prototype pathogen preparedness for pandemics.
Right.
I guess I might just make one comment that I think we're looking at things where humans are, another reason to understand it is because of the way that humans are impacting the globe. When we look at SARS, MERS, avian flu, Ebola, Nipah, Hendra, all of these things, they're all zoonoses, really. They're all from animals. And there's also bat and porcine coronaviruses that are known to infect human cells. So there's really a lot of what sort of, I think Ralph Barrick probably coined the term, pandemic-ready viruses that we know about, in fact. And we haven't seen them in humans yet, but we know their capacity to infect human cells. So I think there's a lot about that. In addition, our intrusion into ecological niches, these viruses evolved really independent of humans or else in small niches around humans, but they didn't have anything to do with us. And most of them have adapted readily. Most bat coronaviruses, or all of them that are known, really don't cause disease. And so it also gives us, it's important for us to look at these because it gives us insight into what our impact is in the rest of the world, not just the impact of viruses on us, but our impact on viruses and their natural hosts, and devastating potential diseases like infection of large primates, infection of mucid animals. So I think there's lots of other reasons to study this. And like Barney said, it's knowable. It's something we can know and something we can do if we pay attention to it.
Well, coming back to that point, I think, Dr. Graham, that you make about a family of 26 viruses. I mean, you actually started out your work and your research exploring the development of vaccines for HIV and RSV, viruses that are prevalent in many different ways. And what was a takeaway, I think, from that research for you that informed the work that you've now done on COVID-19? How can we see those links between past research and future and current research?
It developed a life of its own. It's even got its own division within NIAID, a division of AIDS. But originally, at its root, it was a zoonotic emerging infectious disease that was a regional problem that was unrecognized and became global. And so the way HIV has impacted my work on coronavirus is that it is the vehicle by which I got to NIH to join the Vaccine Research Center. And the work on HIV vaccines, not just by us, but by the world, has generated these new ways of thinking about vaccinology and new technologies and structure-based vaccine design and things like that that have allowed us to have the tools to see things with much more precision. And it's what made our work on RSV possible, which was defining the structure of a protein on the surface of that virus that's analogous to the spike protein on coronaviruses. And when we captured that structure at the atomic level and were able to see the shape of that original F protein, we realized that that could be a vaccine antigen, as opposed to the rearranged F that is what the virus uses to pull itself into a cell. And so that insight into what we call class 1 fusion proteins on envelope viruses is what led us to say, because that work was going on right at the time MERS coronavirus was breaking out in the Middle East. And so, and as Jason McClellan was moving from the VRC to his first faculty position, we said, let's work on coronaviruses together and see if we can transfer that learning on that envelope virus to this other envelope virus and solve the spike protein structure and go from there.
So it's a long story of connectivity across those studies of different viruses in many different projects. And I think that leads me to reflect on Dr. Dennison's work, here at Vanderbilt. And why don't you share with us what it's like to work for so long on a virus that only a small group of people studied? I know I can remember hearing some of your colleagues here rib you about, why don't you study an important virus, Dr. Dennison? And what kept you going? And how'd you secure research when people thought you were working on an obscure virus?
Well, I suppose you might ask my wife, Laura, what was it like to be living? Barney would often, because many people don't know that Barney Graham lived on Barton Avenue just across the street from me, and Kathy Newsel just lived right down the street from us. And so we were close neighbors, and Barney's kids babysat for mine, and we were good friends as family. So he would often reassure Laura that it was going to be okay when I was not so confident that it was going to be okay. So a sort of indefinite uncertainty about whether you could get support. So it was real. I've told a story about being on vacation in Florida in 2003 with my wife, where I'm basically saying, I just don't think I can keep doing this with a murine model coronavirus, even though I found the questions to be profoundly important. And getting a call from a colleague at Vanderbilt saying, hey, have you heard this outbreak of this disease in Hong Kong is a coronavirus? And that was SARS. But I think getting back to your question, you have to be driven by multiple things, and I'd say Barney and Kathy have done this too, because they've seen the challenges associated with getting ongoing, continuous support for vaccines. For getting support for RSV, which only infects children, it's very hard to get the attention and keep that focused attention. But for us, it was I guess the challenges of funding were apparently at least minimally exceeded by the questions that the virus posed. Why did such a large and complex genome and replication strategy survive evolution? How did coronaviruses find their way into so many species? Because we knew that from the mammalian ones that cause devastating diseases in domestic animals. How can we find, when we kept discovering one protein after another that we didn't think exists, the genome kept getting bigger as people discovered how large it was. So I guess whenever I felt discouraged, I also had students, and I wanted to be optimistic for them. And we just managed to keep going enough. And then I guess in some ways my career was rescued by pandemics or pre-pandemics with MERS and SARS that allowed the world to recognize the potential importance of these. And so we've had to keep a long horizon, a very long horizon, and we've had to be predicting things that people didn't want to hear based on what we knew. Right. I think those are the real reasons. And I guess the message is, to me, is I think it's important to have a long horizon. And when you look at Dr. Newsel's work and Dr. Graham's work, they've had very long horizons as well. Barney, I know, was talking about vaccines for RSV when he was here in the 1990s and trying to find vaccines for that. That's a very long and difficult horizon. And you have to take stock of that, and you have to keep thinking about it. I guess the final comment is, I guess there's an advantage. I think of our work as being like an index mutual fund. It rises slowly. It's not dramatic. Not very many people find it interesting, but I think it's better to be that than a growth stock sometimes, where you can have the time for your students to actually explore really critical, novel ideas, and they have the time to develop that without the concerns about that being done by 20 other people at the same time. So there are advantages to that kind of work as well.
Well, we're grateful for your persistence and your resilience, and having all that history and all the research and data that you collected over time to really impact what's happening right now. I think that leads and kind of threads back to a comment Dr. Graham made about biomedical research funding, and how biomedical research is funded, and some could say, are we actually supporting the right research efforts? And I think either, both Dr. Graham and Dr. Nuzel could comment on that from their independent perspectives. Do you want to start
Barney,
off,
After you, Kathy.
Yeah, I'm happy to start. Just to echo some of what Mark said, which we'll get to my comments, is that as Barney said, if you were beginning this profession, if you were an early career investigator in infectious diseases in the 80s, everybody was studying HIV. Everybody but Mark. I actually chose to work with Barney on RSV and with Dr. Peter Wright on influenza viruses. Well, influenza now is all over the newspapers, and we know about pandemics. But even back when I started working on influenza, people said to me, why do you want to work with a cold virus? Because it really wasn't until we had those avian outbreaks, in the 2000s, and people started to talk about the pandemics, that anybody paid attention to influenza, let alone coronavirus or RSV. Which really brings me to what has driven me in my career, is that a lot of decisions that we make every day about vaccines and drugs are market-driven business decisions. And, does a company decide to make all this investment to take a vaccine to licensure or a drug to licensure? That is a business decision. We have to have science and public health driven investments. And that means investments that it's not obvious that, you're going to be able to sell a vaccine at the end, that it's not obvious. And that's where really these investments in science and the investments in funding come in. Because again, our natural capitalist business model is not going to take risks in the same way that we can take risks in science and public health to pursue these different avenues that we've all pursued that have taken decades to pay off. In fact, I would argue that, these mRNA vaccines, which others can talk about, they've been around for a long time, but we never had a business case to bring them to an approval stage the way we did with this pandemic.
Mm-hmm.
Yes, so I can say a few words about something I tell students that are interested in vaccinology, and that is you have to be a person who enjoys delayed gratification, and that if you want to make a vaccine, you got to start early in your career because it often does take decades. And so, but the other thing that's already been mentioned is the connectedness of biology. And one of the privileges I've had in my life and career is that I can study biology and just try to learn about biology. And all of my questions are pretty much biology driven. I started out as a physician. I was chief resident twice in the Vanderbilt system, and I was very much identified. I still identify as a physician, but over the years, I really now identify more as a biologist. And that ability to have a long-term horizon for big projects like the prototype pathogen approach to pandemic preparedness, I envision gaining the knowledge we would need to deal with all these 26 virus families, taking a large portfolio of grants and intramural work, 10 or 20 years to accomplish. And it's a long-term project. It doesn't fit into a political election cycle or a grant cycle. It is a long-term process. And so I've been very fortunate in my life to be able to continue working on projects with a long-term horizon. And that's what it often takes for vaccines. It's a little bit of an exception to that during this last year, because one of my fellows, who is young and just starting her career, Dr. Kizzmekia Corbett, has been involved in this, and all of a sudden she can be associated with a vaccine that's already been approved after a year. And so that rule of thumb wasn't true for her, and she's become really a science celebrity over this just because of what's happened. And so this has been a very unusual year, but I think in general, it is something you're going to have to spend a long time on to really find a biological intervention for disease.
No, I think that the acceleration over the last year also shows what the application of new people coming in, and people who may have been studying CRISPR, or they may have been studying other mechanisms for docking of molecules, suddenly can apply this, and we're seeing this sort of critical mass of energy and driving it. For our purposes, I always worried that we would be the ones called before Congress because, Dr. Dennison, you've been working on a mouse virus of a family of viruses that only cause colds in humans, and the NIH has been funding your work for the last 15 years. What can you tell us about any implications for how this might impact human health? And I would have said, well, now I can say, well, give me 30 years and I'll tell you. And that's hard now because I think the stock model that I said is one that there's this expectation for rapid results, high impact publications. Our work with remdesivir and develop of remdesivir really wasn't fundamentally about antivirals because there was zero market for coronavirus antivirals except potentially in animals, and that's a different whole story. But it was really predicated on identifying an enzyme and a function that we wanted to try to bypass and get around so we could kill the virus with nucleoside analogs. And so the work we did for 15 years was around that enzyme, and the work we did for 7 years was around using that compound really to try to inhibit the virus and understand its fundamental biology. And it just happened that all those publications happened within the year right at and right around the time of the pandemic, which made it possible to move that rapidly into FDA approval as the first antiviral. But it wasn't predicated on that. It was predicated on a long-term understanding of the biology of the virus. So we have to fund that. So my point is we have to fund it. And we have to keep doing that in the different virus families. Now we can't ignore and we can't abandon Zika. We can't abandon the flaviviruses and the alpha viruses and the picornaviruses because suddenly all the money is in coronaviruses. That's exactly what I experienced my whole career. We cannot do that now.
Yeah, I think you make a strong argument for long-term investment and a really broad view of how to approach this critical aspect of science and medicine. And we're hearing lots of discussion in the news about all the new SARS COVID-2 variants that are arising, and will the efficacy of the vaccines be monitored, and how will we know whether or not the current vaccines are effective against those? Is there a way we can proactively address the issue of maybe vaccine failure in regard to some of these new variants? I think these are questions that we're hearing, and it'd be great to hear your thoughts on this particular issue. Dr. Dennison, do you want to pick it up again? And
I will. I'll let Barney start with it, and
okay.
Kathy start with that one because they're. Barney's a deputy director at the VRC, and Kathy is one of the world national leaders in the implementation of vaccines. So I think they'd do great at this.
Well, the variants are getting a lot of attention, and despite the enzyme that Dr. Dennison discovered that does editing for coronaviruses, that enzyme is not doing a great job in SARS-CoV-2, and in part because it's been infecting so many people and it's so widespread, it's had so many chances to adapt. It's not surprising that this RNA virus is changing. In fact, it was expected that it would change, and I think a lot of the changes so far have just made it easier to infect a cell or easier to transmit or easier to survive. And so over this last year and a half, it's made 9 different amino acid changes out of 1,273 in the spike molecule. So it doesn't seem like that many, but some of them are in critical places that affect antibody binding. And antibody is our main weapon here, I think, in terms of immunity against the virus. So this is being tracked with extreme precision, and people are looking for new variants in almost every case of coronavirus now. And as those changes emerge and are tracked and followed, we're also able to adapt our assays to ask, will our vaccine sera that has the antibodies in it still neutralize or block those kinds of viruses? And so we can track this in real time. And just as we developed the original vaccine that was effective, this technology, these new rapid manufacturing approaches allows you to very rapidly adapt the vaccine. So 2 or 3 days ago, a new study was started with an mRNA vaccine that encodes the new strain that people are concerned about that came out of South Africa. So these, the tools that we have now not only allowed us to respond, but it allows us to adapt just as the virus is adapting in real time. I'm pretty confident that we can either keep up or get ahead of the virus. And the real important thing for people to hear is that these viruses will continue to adapt and continue to evolve every time they get a new chance to infect a person. And so for all of those people out there who are hesitant or lack confidence in the vaccination process, I just hope that they will continue to avoid being infected, because you would not want to be the person on Earth who grew out the next variant virus because it adapted to infecting you. And that virus now escapes our vaccines and therapies that we're so lucky to have. And so that is an important thing for people to understand, that each time an infection occurs, there's a chance for a new adaptation that could escape our effective vaccines and therapies.
Yeah, and I think just to emphasize, investments in basic science and technology are going to go a long way in terms of fighting these variants. Influenza is another virus that changes frequently. We've learned a lot about priming and how your first infection matters and subsequent infection matters. And as Barney said, we have a lot more tools now. I think the other point, and Barney and Mark are virologists and biologists, and while I spent time in Barney's lab, I'm really an epidemiologist, and I consider myself a trialist and a population scientist, and we have to think about control of this virus in that way. And these variants, in fact, were emerging in the absence of vaccine in areas where you just had unmitigated viral replication and no control of this virus. So to emphasize what Barney said, the answer here is certainly to roll out the vaccines we have now as quickly as possible, that this has to be a global response. Even if we vaccinate everyone in the US and variants are emerging in other places, we have to have a global response. We have to have a global plan. We have to have a global vaccination plan. We have to have a global surveillance plan in in order to get this under control. And then finally, if you look at the modeling and particularly the transmission modeling, again, the more people that are vaccinated, the more we are all protected. Because this isn't tetanus, which lives in the ground and we each individually get exposed to that spore. This is a virus that's transmitted from person to person. So if I'm protected and the people around me are vaccinated, then I am even more protected. And we just have to keep remembering that. We focus on this individual efficacy in a clinical trial, but in fact, this is really a transmissible population level control that we're looking for.
Yes, and I think it's important also for maybe us to have a discussion about the possibility of the development of a universal coronavirus vaccine. That might follow up in terms of this variant discussion, but I know there's been lots of work that's been done on the development of a universal flu vaccine, but Dr. Nuzell, what do you think in regards to a universal coronavirus vaccine? Are efforts already underway? Is that a possibility on the horizon?
Yeah, well, again, I'd love to hear Barney and Mark's comments as well. It's just a different way of thinking. We've clearly been highly successful in responding to a vaccine that's very specific to this virus and that S protein by having these unprecedented vaccine development timelines and rollout that we've seen in the past year. But again, as you've heard all of us say, there have been 3 emerging coronaviruses with severe effects on humans over the last 20 years. You used influenza. There have been, multiple influenza pandemics in the last 100 years. So it is absolutely appropriate while we continue to improve and move forward with these highly effective vaccines that we do start to think about, coronaviruses as a whole, as a family, how we might target them, how we might be ready for that next coronavirus. And I would say not just vaccines, but, Mark's work with antivirals has been critical. We absolutely need an oral, easy to take antiviral that works against multiple coronaviruses, and using that with a vaccine could really go a long way, again, to both early control and control in the situation we are now with a true pandemic.
I guess Barney's, I've been thinking about pandemic preparedness, but I got to come back to the biology, not to defend my virus, but virus family, but to talk a little bit about their evolution. It's like it's a family, and every member is dysfunctional in their own way. In some ways, I like the RNA technology because, in fact, you don't have to think, and this may be antithetical to what Barney would say, but you don't have to think universal. What you can do is you have a technology and a methodology that allows you within days to be generating a vaccine that would be efficacious against a very specific virus. I will say a little bit about the biology. We did discover proofreading. What we show is that coronaviruses make about 100 times less mutations than other RNA viruses. But I think we have to be sure we're comparing the right things. They still make about 1 million times more mutations than our DNA-based organisms do. So they may be better than other RNA viruses, but they and then coronavirus is a little bit, I call it, of being able to have their mutations and eat them too. We've always demonstrated over the years that under stable conditions, they don't change at all. But if you introduce a selective variable, an evolutionary pressure, an antibody, an antiviral, a vaccine, a mutation that's dangerous to the virus or that damages the virus, it can adapt in a matter of minutes, days, or hours, days. Okay, they are profoundly good at it. And I think that's why coronaviruses may be really almost ubiquitous in mammals and across species, because they seem to have this capacity for absorbing mutations, of changing when they need to and being stable when they need to. And so for those of us that have worked in the field a long time, this kind of variation it's scary. But it's not a surprise. And so we need to be really understanding those determinants if we're going to look at anything that would be universal toward vaccines.
It turns out that we had some pan-coronavirus vaccine concepts that we were ready to put into animal studies before this pandemic started. And we've had to set them aside while we're dealing with the current pandemic. But there are 4 endemic coronaviruses that circulate among humans every year. 2 alpha coronaviruses and 2 beta coronaviruses. And so if you combine those with the 3 that have emerged, there have been 7 human coronaviruses that cause disease. And after we were able to solve the spike structure of a human coronavirus back in 2016 and then learned how to stabilize it in 2017, we started working with companies, not just to make vaccines, but to use those same tools and reagents to discover cross-reactive antibodies for coronaviruses. So we worked with Abcelera and gave them the probes to look for B cells that could bind OC43 and HKU1 at the same time, for example. And so we can now not only isolate cross-reactive antibodies that could be used for treatment, but we can also build what we call nanoparticle vaccines, where you can self-assemble proteins that would then display spikes, and you can have multiple spikes on the same particle. You can have a cocktail of spikes, and that's where we were when this pandemic started. And it isn't out of the question to have a relatively pan-coronavirus vaccine. And now there's a lot of other examples. And I did give a talk last week on this subject, and the title was Pan-Coronavirus Vaccine Development Following the Influenza Playbook, because that is our task in influenza, is figuring out how to overcome the genetic variation of influenza and adapt to the pre-existing immunity, avoid the immunodominance. And we have developed a lot of interesting new ways of immunizing for influenza that can do those kinds of things. So we know how to make coronavirus vaccine antigens now, and we are learning new ways of avoiding or dealing with this genetic plasticity and antigenic variation from influenza work. So I think the technologies we have are so stunning to me. I couldn't have imagined what we could do 30 years ago. And so I think we do have the tools to deal with these things.
So
It's a matter of
dive.
investment.
Yeah. Let's dive into that reference to technologies. And I know this is a messenger RNA based vaccine strategy for delivery of the vaccine. What makes it so effective, especially compared to other methods?
So this should definitely be a Barney first.
Well, we chose mRNA for this project on pandemic preparedness that we started in 2017. We started a project on paramyxoviruses and coronaviruses with Moderna, and we chose mRNA in part because it was fast. But we also chose it because it's a good way to induce an immune response, and it can create the kind of immunity that is able to mimic an actual virus infection without having the infection. And so there's a number of nice things about RNA. Historically, vaccines have been used either a live attenuated virus where you get a mild infection, or a whole inactivated virus where it's really just a bag full of proteins, or just make the protein and give the protein. Over these last 30 years, people have developed ways of delivering the gene that encodes or provides the template to make the protein in the body itself. So now our cells become the factory for making the vaccine antigen, the protein. And you can deliver that with an adenovirus or a pox virus vector, or you can deliver it with nucleic acid like DNA, or even better, RNA, because RNA is the simplest, most elemental chemical form of being able to make a protein. And a single strand of RNA can encode this protein, and the cell makes it, it folds it the right way, it produces it in the way that it would be produced in a virus infection, only you're not infected. So mRNA has a number of nice properties, and it engages not only the antibody arm of the immune system, but it creates T cell immunity, what we call CD8 T cells that are good at clearing virus-infected cells. So it embraces all those elements of the immune response that we like. And it doesn't have anti-vector immunity problems like some of the vectors. So you can, if you're giving it as an adenovirus or a pox virus, there's only a limited number of times you can do that before now you have immunity to the vector, the carrier. Well, you can give RNA over and over and over. But the reason we chose it in the beginning is because it's fast. And we wanted to pair our precision antigen design, or the atomic level structure guided antigens that we thought were the right thing for the immune system to see, with something that could have rapid delivery. And that's the reason we chose it in the beginning.
Well, thank goodness it's fast. I can say that. Thank goodness it's fast and has been so fast. Now there are questions, and this might be one especially for the more general audience, in terms of the questions about why do some of these RNA vaccines have to be stored at such ultra cold temperatures, minus 90 degrees Celsius, whereas others can be in a refrigerator. What's that reflective of, and how can we help people think about that?
You want me to go first?
Sure, and then we'll...
You bet.
You could probably answer this question yourself, Dr. Wente, because you are a cell biologist and you know how fragile RNA is. Dr. Nuzzel knows how fragile RNA is, because that was our first big paper together, where we were trying to get the RNA out of the lung to figure out what kind of cytokines were being produced. And it was horrible because RNA is not very stable. But what's happened over the last 15 years is, because of technologies that were initially developed around small interfering RNAs, these little pieces of RNA that have been discovered that end up regulating our gene expression and all sorts of things. So people were trying to turn these small RNAs into drugs. And they worked for 10 years figuring out how to stabilize the RNA and keep it and make it as a product, mostly through the development of these lipid formulations that can encase the RNA and protect it from degradation. And so both Pfizer and Moderna use a mixture of 4 different lipids, and one of them is pegylated, and that lipid formula forms a little bubble around the RNA and protects it against the elements, but it also helps deliver it into the cell. So, but as Kathy said earlier, RNA has never been scaled up before. Most of these companies have been, except, as they evolve from the small interfering RNA particle projects, they are doing cancer vaccine work. They're mostly addressing using RNA vaccination for cancer and making personalized vaccines that are in very small batches. And so nobody's ever scaled this up before. And so that's the reason the storage temperatures have not been brought down. Now, eventually, we will be able to package RNA in a lipid nanoparticle and freeze dry it and let it sit at room temperature, and it'll be just fine. It's just that what's called process development, part of vaccine manufacturing, has not really been done into a large scale like you would need for this kind of a vaccine program. And that's all been done kind of building the bus as you drive.
So I just want to give a shout out to viruses here. And what you're describing is exactly what an RNA virus is in some senses, right? So I want to be clear that these vaccines are not viruses, because I spend a lot of my time every day talking to people about that. But that, the coronaviruses encode the largest and most complex genome RNA molecule known, that can infect people, and are encapsulated in a lipid particle with proteins and a few other carrier molecules to get into a cell and then reprogram the entire cell. So I think we've learned to sort of replicate what nature had done in some ways, to use it against the viruses, which is stunning to me. It's just really, sorry, as a biologist, I hesitate, but I use the word miracle. It's a miracle in terms of the speed that this can be done, and I only say that predicated on work of people like Barney to identify the structure of the protein, and the people who've worked on this RNA biology. And that's another area of what you would maybe call obscure research, siRNA technology, RNA as a delivery vehicle for antigens. Those were not areas that would have fundamentally drawn a lot of attention at the national level.
So how easily do you think that this mRNA vaccine technology can be adapted to new viruses? I think you made reference to that, but if it can be easily adapted, will the same rigorous testing need to be done, or will there be other aspects of like the clinical trial testing that can be streamlined? Maybe Dr. Nuzil, would you like to address that?
Sure, and I think Barney alluded to this. When you have a genetic sequence and you can make changes to that genetic sequence to adapt to a variant virus or a new virus, but use that same lipid coat and the same manufacturing process, then yes, that is really the beauty of these mRNA vaccines, which is how quickly they can be manufactured. And the FDA did put out guidance a couple weeks ago in regard to these variant strains, that actually you wouldn't need to do another huge clinical trial, but you could in fact do something we call bridging to the large clinical trials that we've already done. So we could see if the immune response looks similar, for example, against a new variant strain, and then that could be used. So a much smaller study in humans could be used to license these new vaccines. Now, of course, we can do more in-depth studies in animals, and there was a lot of supportive work done in laboratory and animals for the current vaccines. But it would be more similar to what we do with an influenza strain change, where we don't do big new clinical trials every year for a new influenza vaccine.
Could I just make one additional comment, and that is mRNA is a great delivery vehicle, and we've shown that this year. Now it's been in tens of millions of people, and it will be used again, but it will not work if you don't deliver the right thing. And so I just want to emphasize the importance of making the right thing. For instance, if it was an RSV vaccine, if you made an F protein that was flipped into the post-fusion conformation, it would not work as a vaccine. Unless that protein stays in the right shape, it won't make the right kind of immune responses. So I just want people to remember that it doesn't matter how fast you go if you don't do the right thing.
Powerful, powerful point. Absolutely. Mm-hmm. Dr. Dennison, were you going to add something?
No, just that every virus, and just, since I've only studied one virus family my whole life, I just can comment that, this one is shocking us, and me, especially in its broad tropism in humans, meaning the kinds of tissues it can attack and the kinds of places it can go, which differs from what SARS did and differs from how MERS has manifested in some ways, and certainly differs from the other human coronaviruses. So I think that the challenges of the presentation and vaccines is that you have to understand what the virus is doing as well, and what those proteins can do, and what the implications of it are. So I think we won't avoid scrutiny with new vaccines, but it will definitely accelerate getting those into testing very, very rapidly. And I'm very excited about that for the future of so many different viruses. And Barney won't emphasize the importance of the work that their group did, that he led, in terms of this novel mechanism for stabilizing that protein, which is so broadly applicable across viruses. And this would not be happening now if that hadn't happened, because the 3 vaccines we're all talking about all are using that same approach for stabilizing the spike protein to express it in ways that give us good immune responses.
Yeah, the key part of this entire puzzle being that critical role of that protein. So we're nearing the end of our time here, and I want to have us have a chance to have a discussion amongst all 3 of you about what do you think we've learned the most in regards to from this pandemic that will help us be prepared for what is likely to be the next pandemic, and the key steps that we should take. What can we really drive home from this?
Yeah, I'm happy to start. There was an article just this week that talked about the cost of this pandemic. We know the cost in human lives, which has been enormous. And, it's into the trillions of dollars in terms of what this has done for our economy. So I hope we scientists no longer need to justify investments in pandemic preparedness, because it's quite clear what accelerating vaccine development has done and what it has saved. We've all made the point that investments in science and technology absolutely enabled us to be where we are. And they're not necessarily investments in specific coronavirus science and technology. Some of it is, but again, Barney and Mark and we've all made the point of how improved assays and improved ways of delivering vaccine, protein conformation, these scientific advances. I think the second issue is the investment of resources. We've never been able to have the resources to work in parallel the way we have worked this past year in terms of moving these vaccines forward. We were able to do large trials. We were able to start process improvements and manufacturing vaccine before we even knew if this vaccine was going to work. And without those resources and public-private partnerships, we couldn't have done that. And then to me, the third issue has been partnership. Barney, Mark, and I, we were saying how we lived in the same neighborhood. We've seen each other more in the last year than we've ever seen each other before, because, the US government, the World Health Organization, brought many partners to the table. We had manufacturers, we had the FDA, we had the NIH, we had independent experts like Mark, we had, experts in clinical trials and all the trial networks that we've invested in over the last 30 years. And everybody sat around the table and they shared lessons learned and they worked together. And so, to me going forward, it's investments in science and technology. It's making sure we have the resources. And then it's this partnership model that will really enable us to have better pandemic preparedness.
Dr. Graham? Mark?
Barney, go ahead.
Okay. Well, I'll start, and I hope what we've learned this year is how connected we are around with people all over the world, and that a problem anywhere is a problem everywhere, and that regional diseases can become global diseases if we don't take care of them regionally. And so this idea that we are all connected and interdependent, and viruses don't pay attention to geographical borders. And now you can have a virus on one side of the world be on the other side of the world in just about 12 hours on an airplane flight. So this idea of interconnectedness, and I think is what I hope the world remembers, because I think we need a 194 country accord, just like we have for climate change on emerging infectious diseases, so that we can really wrap our heads around what needs to happen to prepare for the next thing. And I think another testament to the technology advances is that on the WHO landscape slide, there is over 250 coronavirus vaccine projects, okay? And 70 of them are in clinical trials. And I don't think it makes sense that 250 different vaccine groups are making a vaccine to the same one virus. And that if we could divide them up into groups of 10, we could have 10 vaccine groups working on each of these 26 viral families that we're worried about in the future. And so we now have the technology, we have the capacity, we have global capabilities, and we need to harness that with a more of a global approach to this problem.
Well, I just want to comment on one aspect that maybe it doesn't come up on the top of these types of conversations, and I wish we could start over and have a conversation around this as well. And that's the issue of, workforce is such an uninteresting word, but what I mean is, you asked me at the beginning about sort of working with an obscure virus. I have to say that I had been pondering how my lab, when I came to the conclusion of my career, would shut down and how I would get rid of all of these mouse hepatitis virus mutants and resources and reagents and those SARS and those of MERS, because there wasn't an interest in trainees, and that people going out, students and postdocs going out, would choose to other fields of either of virology or of microbiology, or not even within biomedical research directly, because they saw how hard it was for me to get that work done and support it. And so I would argue one thing we need to learn and we need to carry forward if we're going to be prepared is that technology is meaningless. Epidemiology is meaningless unless there are people who are trained and committed and view these as viable careers. So we're at Vanderbilt, we're talking about this, right? And we're talking about these important steps. So how do we make sure that there are stable enough pathways that people can explore the crazy ideas that lead to the new technologies? How can we build methods within institutions like Vanderbilt for tenure and other places that allow for people to be rewarded for team efforts and massively parallel research? Rather than individual research, rather than holding back information and being able to share it openly. The preprint servers have done that some. There's a meeting I'm on every week now with coronavirus experts. It's a big group of people who are sharing data ahead of time that's not been published yet. Well, that's that. I guess I just want to leave it with that. Our hope really doesn't lie in just the technologies. It lies in the trainees and the people and the recognition that we need diversity and we need those vast creative resource that allows people to take time and take chances.
Is it okay to just make one more comment?
Please,
No,
absolutely.
Mine was perfect. Mine was perfect.
But
You
they
gotta
all
stop
have
there.
to be one.
Unfortunately, this comment could lead to another 3-hour discussion, but that idea of connectedness and interdependence that I was talking about, I wanted to also say that this pandemic has uncovered social and medical and every other kind of inequity in our society. And I think that the idea that, essential workers being more exposed to virus puts them at greater risk. And what we call essential workers often have lower salaries and have less ability to connect to digital world and healthcare options and even vaccine options. And so I hope the other thing we've learned from this process and our interconnectedness is that we can solve some of those inequities. It's another thread of my life that has come together in this big event. And this, Mark mentioned the importance of diversity, not only in access to healthcare, but in innovation and bringing some of these new ways and perspectives into the lab and solving some of these societal problems. I think this pandemic has revealed the depth of some of our disparity that needs to be corrected.
Mm-hmm. Well, that's an incredible set of really powerful lessons learned from not only your careers and the long road that you've taken to get to where we are today, but also especially from what we've learned over the last year and a half. And I think I'm really hopeful that our audience will reflect on those powerful lessons learned and embrace interconnectedness and embrace partnerships and embrace really stepping forward to address these inequities and issues that need our investment and our attention. But you give me hope. The three of you, in terms of how you've really turned your forces for such greater good, you truly give me hope. And I want to thank you for all the work that you've done and that you've been doing, and for being willing to come back to Vanderbilt virtually to share with our audience here today. Thank you so much. And it's been wonderful, wonderful to share the screen with you. Thank you.
Thank you, Susan.
Thank you.
Thanks for having us.
Thank you so much. This has been a wonderful conversation. I wish we had more time to talk, but we must let you get back to your important work. Thank you, Chancellor Diermeier, Vanderbilt University Medical Center, and the Alumni Association for bringing this one-of-a-kind event here today. Finally, thank you to the entire Vanderbilt community for being my home in all my academic and leadership roles for all these years. It's been my profound honor to help advance our mission for education excellence and societal impact, which is what fuels the remarkable breakthroughs like those we've discussed today, and which promises to continually take us to new heights of enabling human potential. I'll now hand it back to Chancellor Diermeier to close the session.
Thank you to Provost Wente and our panelists for that enlightening discussion and for your tremendous humanitarian contributions. And thank you for reminding us that amazing scientific discoveries do not happen overnight. They require the steadfast pursuit of basic research, collaboration across multiple fields, and institutional resources to collect and disseminate vital knowledge. Vanderbilt is proud to have supported the decades of work that led to some of today's most prominent COVID-19 treatments and vaccines. We also know that much more work lies ahead. In our increasingly connected global environment, pandemic prevention will be a critical pursuit for many years. Thank you again to the Vanderbilt Alumni Association and to Vanderbilt University Medical Center for hosting today's events, and thank you all for joining me at the Chancellor's Lecture Series. I look forward to seeing you again soon.